Welcome back. We are on Chapter 4, Section 3, non-proteobacteria, gram-negative bacteria, and phototrophic bacteria. Learning objectives. Describe the unique features of non-proteobacteria, gram-negative bacteria. Give an example of a non-proteobacteria bacterium in each category. Describe the unique features of phototrophic bacteria. Identify phototrophic bacteria. The majority of the gram-negative bacteria belong to the phylum Proteobacteria, discussed in the previous section. Those that do not are called the non-proteobacteria. In this section, we will describe three classes of gram-negative non-proteobacteria: the spirochetes, the CFB group, and the Planctomycetes. A diverse group of phototrophic bacteria that includes protobacteria and non-proteobacteria will be discussed at the end of this section. Spirochetes. Spirochetes are characterized by their long, up to 250 micrometers, spiral-shaped bodies. Most spirochetes are also very thin, which makes it difficult to examine gram-stained preparations under a conventional bright-field microscope. Dark-field fluorescent microscopy is typically used instead. Spirochetes are also difficult or even impossible to culture. They are highly motile, using their axial filament to propel themselves. The axial filament is similar to a flagellum, but it wraps around the cell and runs inside the cell body of a spirochete in the periplasmic space between the outer membrane and the plasma membrane. And that's illustrated in this figure. Several genera of spirochetes include human pathogens. For example, the genus Treponema includes a species, T. pallidum, which is further classified into four subspecies: T. pallidum pallidum, T. pallidum pertinue, T. pallidum caradium, and T. pallidum endemicum. The subspecies T. pallidum pallidum causes the sexually transmitted infection known as syphilis, the third most prevalent sexually transmitted bacterial infection in the United States, after chlamydia and gonorrhea. The other subspecies of T. pallidum cause tropical infectious diseases of the skin, bones, and joints. Another genus of spirochete, Borrelia, contains a number of pathogenic species. B. burgdorferi causes Lyme disease, which is transmitted by several genera of ticks, notably Ixodes and Amblyomma, and often produce a bullseye rash, fever, fatigue, and sometimes debilitating arthritis. B. recurrens causes a condition known as relapsing fever. Appendix D lists the genera, species, and related diseases for spirochetes. Cytophaga, Fusobacterium, and Bacteroides. The gram-negative non-proteobacteria of the genera Cytophaga, Fusobacterium, and Bacteroides are classified together as a phylum called the CFB group. Although they are phylogenetically diverse, bacteria of the CFB group share some similarities in the sequence of nucleotides in their DNA. They are rod-shaped bacteria adapted to anaerobic environments, such as the tissue of the gums, gut, and rumen of ruminating animals. CFB bacteria are avid fermenters, able to process cellulose and rumen, thus enabling ruminant animals to obtain carbon and energy from grazing. Cytophaga are mobile aquatic bacteria that glide. Fusobacteria inhabit the human mouth and may cause severe infectious diseases. The largest genus of the CFB group is Bacteroides, which includes dozens of species that are prevalent inhabitants of the human large intestine, making up about 30% of the entire gut microbiome. 1 gram of human feces contains up to 100 billion Bacteroides cells. Most Bacteroides are mutualistic. They benefit from nutrients they find in the gut, and humans benefit from their ability to prevent pathogens from colonizing the large intestine. Indeed, when populations of Bacteroides are reduced in the gut, as often occurs when a patient takes antibiotics, the gut becomes a more favorable environment for pathogenic bacteria and fungi, which can cause secondary infections. Only a few species of Bacteroides are pathogenic. Bacteroides melanogenicus, for example, can cause wound infections in patients with weakened immune systems. Planctomycetes. The Planctomycetes are found in aquatic environments, including freshwater, saltwater, and brackish water. Planctomycetes are unusual in that they reproduce by budding, meaning that instead of one maternal cell splitting into two equal daughter cells, as in the process of binary fission, the mother cell forms a bud that detaches from the mother cell and lives as an independent cell. These so-called swarmer cells are motile and not attached to a surface. However, they will soon differentiate into sessile, immobile cells with an appendage called a holdfast that allows them to attach to surfaces in the water, as in this figure. Only these sessile cells are able to reproduce. So this table summarizes the characteristics of some of the most clinically relevant genera of non-proteobacteria. So these are all gram-negative, phototrophic bacteria. The phototrophic bacteria are a large and diverse category of bacteria that do not represent a taxon, but rather a group of bacteria that use sunlight as their primary source of energy. This group contains both proteobacteria and non-proteobacteria. They use solar energy to synthesize ATP through photosynthesis. When they produce oxygen, they perform oxygenic photosynthesis. When they do not produce oxygen, they perform anoxygenic photosynthesis. With the exception of some cyanobacteria, the majority of phototrophic bacteria perform anoxygenic photosynthesis. One large group of phototrophic bacteria includes the purple or green bacteria that perform photosynthesis with the help of bacteriochlorophylls, which are green, purple, or blue pigments similar to chlorophyll in plants. Some of these bacteria have a varying amount of red or orange pigments called carotenoids. Their color varies from orange to red to purple to green, as depicted here, and they are able to absorb light of various wavelengths. Traditionally, these bacteria are classified into sulfur and non-sulfur bacteria. They are further differentiated by color. The sulfur bacteria perform anoxygenic photosynthesis using sulfites as electron donors and releasing free elemental sulfur. Non-sulfur bacteria use organic substrates such as succinate and malate as donors of electrons. The purple sulfur bacteria oxidizes hydrogen sulfide into elemental sulfur and sulfuric acid and get their purple color from the pigments bacteriochlorophylls and carotenoids. Bacteria of the genus Chromatium are purple sulfur gamma proteobacteria. These microorganisms are strict anaerobes and live in water. They use carbon dioxide as their only source of carbon, but their survival and growth are possible only in the presence of sulfites, which they use as electron donors. Chromatium has been used as a model for studies of bacterial photosynthesis since the 1950s. The green sulfur bacteria use sulfide for oxidation and produce large amounts of green bacteria chlorophyll. The genus Chlorobium is a green sulfur bacterium that is implicated in climate change because it produces methane, a greenhouse gas. These bacteria use at least 4 types of chlorophyll for photosynthesis. The most prevalent of these, bacteriochlorophyll, is stored in special vesicle-like organelles called chlorosomes. Purple non-sulfur bacteria are similar to purple sulfur bacteria, except that they use hydrogen rather than hydrogen sulfide for oxidation. Among the purple non-sulfur bacteria is the genus Rhodospirillum. These microorganisms are facultative anaerobes, which are actually pink rather than purple, and can metabolize fix nitrogen. They may be valuable in the field of biotechnology because of their potential ability to produce biological plastic and hydrogen fuel. The green non-sulfur bacteria are similar to green sulfur bacteria, but they use substrates other than sulfides for oxidation. Chloroflexus is an example of a green non-sulfur bacterium. It often has an orange color when it grows in the dark, but becomes green when it grows in sunlight. It stores bacterial chlorophyll in chlorosomes, similar to chlorobium, and performs anoxygenic photosynthesis using organic sulfites in low concentrations, or molecular hydrogen as electron donors. So it can survive in the dark if oxygen is available. Chloroflexus does not have flagella but can glide, like Cyanophagia. It grows at a wide range of temperatures from 35 degrees Celsius to 70 degrees Celsius, thus can be thermophilic. Another large, diverse group of phototrophic bacteria compose the phylum Cyanobacteria. They get their blue-green color from the chlorophyll contained in their cells. Species of this group perform oxygenic photosynthesis, producing megatons of gaseous oxygen. Scientists hypothesize that cyanobacteria played a critical role in the change of our planet's anoxic atmosphere 1 to 2 billion years ago to the oxygen-rich environment we have today. Cyanobacteria have other remarkable properties. Amazingly adaptive, they thrive in many habitats, including marine and freshwater environments, soil, and even rocks. They can live at a wide range of temperatures, even in the extreme temperatures of the Antarctic. They can live as unicellular organisms or in colonies, and they can be filamentous, forming sheaths or biofilms. Many of them fix nitrogen, converting molecular nitrogen into nitrites and nitrates that other bacteria, plants, and animals can use. The reactions of nitrogen fixation occur in specialized cells called heterocysts. Photosynthesis in cyanobacteria is oxygenic, using the same type of chlorophyll as found in plants and algae as the primary photosynthetic pigment. Cyanobacteria also use phycocyanin and cyanophycin, two secondary photosynthetic pigments that give them their characteristic blue color. They are located in special organelles called phycobilisomes and in folds of the cellular membrane called thylakoids, which are remarkably similar to the photosynthetic apparatus of plants. Scientists hypothesize that plants originated from endosymbiosis of ancestral eukaryotic cells and ancestral photosynthetic bacteria. Cyanobacteria are also an interesting object of research in biochemistry, with studies investigating their potential as biosorbents and products of human nutrition. Unfortunately, cyanobacteria can sometimes have a negative impact on human health. Genera such as microch44part3 can form harmful cyanobacterial blooms, forming dense mats on bodies of water and producing large quantities of toxins that can harm wildlife and humans. These toxins have been implicated in tumors of the liver and diseases of the nervous system in animals and humans. This table summarizes the characteristics of important phototrophic bacteria. We have our cyanobacteria, our chlorobi, our chloroflexi, and of course there are representatives in alpha, beta, and gamma proteobacteria that are phototrophic. Only one of these groups, the cyanobacteria, engage in oxygenic photosynthesis. All the rest of them engage in anoxygenic photosynthesis. And that brings us to the end of Section 3. Join me next time for Section 4, gram-positive bacteria. Until then.